Okay, I think we're good to go. People happy with that? Yes, no, maybe so. Thumbs up. Okay, fantastic. Um, Thank you all for coming today. It's my absolute pleasure to welcome you to the ASAP Polaris OpenADMET Community Blind Challenge Participant Showcase, which is a bit of a mouthful. I'm Hugo from the team at OpenADMET, previously at ASAP. Uh, Really looking forward to hearing from our three speakers today who are going to be showing off the fantastic science and ingenious technical strategies they use to come to the upper echelons of the Community Blind Challenge. But before we get into that, I'm just going to give a little bit of background, maybe for people that didn't get a chance to, or as much of a chance to follow the full saga of the Blind Challenge, kind of how we got here. And also, Cass from the team at Polaris is also going to say a few words. Okay, so first, ASAP. So ASAP provided, at a high level, ASAP provided the data for real-world drug discovery data that was used in the blind challenge. So maybe a little bit about of background about ASAP and kind of how we got here. So ASAP is an academic-led biotech focused around antiviral drug discovery for pandemic preparedness. So pandemics are of huge concern. As we found out during the COVID pandemic, we were caught a little short without kind of advanced assets for pandemic drug discovery. And in the wake of this, the kind of NIH decided to put some money towards advancing antiviral drug discovery for pandemic preparedness. So they funded several of these AVID centers, of which ASAP is one, but ASAP itself kind of grew out of the COVID moonshot. I won't go into the full details of that today. The best way to think about ASAP is kind of as a distributed biotech. So we're focused mainly on three viral families of pandemic concern: the coronaviruses, 5E viruses, and picornaviruses. And we kind of have all of the normal octopus arms of the drug discovery enterprise. So like target validation, target enablement, structural biology, hit to lead, fragment to lead chemistry, lead optimization, and then preclinical development and translation. But kind of instead of being housed under one roof, it's distributed across a variety of academic and industry partners. So it's brought together some really expert people that you can see on the slide, but all really passionate about antiviral drug discovery. So, as I mentioned, ASAP has pursued kind of several targets across these three family of viruses: the coronaviruses, which we all know and probably don't love; the flaviviruses, which are viruses of concern for dengue and Zika virus you may have heard of, and also enteroviruses. And within these viruses, we've kind of pursued multiple targets that are known to be important for virulence. So in SARS and MERS, these are things like the main protease, which is our most advanced program. And in flaviviruses, it's things like the NS2B, NS3 protease. So if you actually go to our website, so this kinds of lists the progression of all of our programs. But if you go to our website, you can kind of click on each of these and you can see the kind of open science that is behind this. So ASAP has a huge focus on open science. That's how we were able to kind of provide all of this data for the challenge. So you can go and you can kind of see like our target candidate profiles, our the structural biology that we've done, a lot of the potencies that we've measured. All of this is interactive, and where we can, except for in late stage lead optimization, we share as much data as possible with the public. So I said earlier that our main protease programs were the most advanced. We actually recently reached a preclinical candidate, which the whole team at ASAP are super proud of. It's been the culmination of many years' work by lots and lots of people. I know many of you are on this call. You don't need me to congratulate you, but I think this is a moment of some joy for many people. Um, but because we have this open science mission, this also presented us with a very unique opportunity where we had data from our lead optimization, which we keep private while it's in that stage for IP reasons. But then we were planning on releasing this data to the public, kind of not like you would do at a large, say, large pharma company. So we had an opportunity where we had data that kind of hadn't yet been released to the public, real drug discovery data that hadn't yet been released to the public that we could run a blind challenge on. So we partnered with the amazing folks at Polaris, which has come out of Valence Labs, which is itself powered by Recursion. Cass can tell me if I've messed that up in any way. to basically run a blind challenge where people can submit their modeling efforts, and then we kind of judge it against a hidden test set of real data. So kind of what kinds of data are we talking about? So ASAP's partnered with some really amazing folks across a whole bunch of data modalities. So the fantastic folks at Diamond Light Source have produced a really large amount of X-ray structural data for us. And through a combination of high throughput chemistry and high throughput screening at Enamine and the Weizmann Institute, respectively, we've done a lot of bioassays, measured a lot of biochemical potencies. And you can actually see like a lot of this data is already in the public domain, and also so are the protocols. So you can actually go and see how a lot of this data was collected, if you like. But this basically led us to phrasing this challenge across the three main data modalities, which is biochemical potency. So the ADMET endpoints as well. So when you do drug discovery for both safety and pharmacokinetics and efficacy reasons, very important to assess your the kind of ADMET performance of your molecules of interest, and also kind of ligand poses. So they're trying to predict the X-ray crystallography data. So across all of these challenges, the participants were given a training set and then assessed on a kind of blind test set, so a holdout set that wasn't revealed until the end of the challenge. So this is kind of just a little quick breakdown of kind of how the challenge progressed on the bottom end of the screen, and also the kind of fantastic diversity that we saw across the participants. So we had a small number of people come in who were just kind of independent ML people, lone wolves that were really kind of passionate about ML for its own sake. Some people that were more that worked for like software companies in the drug discovery space or similar. Some people that worked in industry, so people that worked in the drug discovery industry proper, and the majority of the participants were academics. So about three-quarters were academics. You can kind of see the progression of the challenge on the below little graph. So we started the challenge. Participants submitted their modeling efforts. They were then evaluated. We disclosed a patent, which then allowed us to release the data into the public domain. And we're still progressing through a series of symposiums, of which this is one, up to some final paper, some preparation of a special issue with the Journal of with JCIM, which many of you will have already heard about. So this was also something fantastic that kind of came out of the challenge, was that everyone kind of centralized around the Polaris Discord, and there was really robust scientific and technical discussion around the challenge, but also modeling approaches for ADMET and for all of the different data modalities. And you can kind of see the progression of the number of messages sent over the challenge. So if you haven't already joined the Polaris Discord and you want to learn more about both how the challenge ran and also the continuing ongoing discussion, I'd really highly recommend that you do. So without further ado, I think you probably want to hear less from me and more from the people that did the fantastic modeling that kind of, as I said, propelled them to the upper echelons of our challenge. So we have Daniel from Shenzhen, Alex from Inductive, and Will from VISTEC, who were some of the top performers across the three modalities that I mentioned. And this is roughly how the schedule will run. So Cast is actually going to say a few words about Polaris first, and then we'll pass to Daniel, and then to Alex, and then to Will. So it'll be about 15 minutes to present, followed by 5 minutes of questions. And we have quite a long slot. So afterwards, if people want to use some unstructured discussion time, that's perfectly fine. But please try and stay on mute and save your questions for the 5-minute question time at the end of each participant. Sound good? I think Cass has a few words to say about the Polaris platform.
Yeah, so with some further ado, I'm going to try to keep this very brief. I really don't want to take too much of everyone's time. But I did want to use the opportunity to provide a little bit of context on Polaris. So if you would go to the next slide, please. So Polaris is a broader initiative. We've seen it in the context of this specific competition, but we're actually trying to tackle a larger problem, and that problem is that benchmarking and drug discovery is just inherently difficult. It requires, especially compared to other popular machine learning domains such as computer vision or natural language processing, it's a lot less intuitive. It requires this interdisciplinary expertise or this industrial experience to truly understand the problems that we're trying to solve and the utility of the various solutions that are being proposed. And so that's just a really tough thing to do that very few people can do well. And then at the same time, benchmarks are very important in machine learning research. They don't only inform decision making, but they also inspire entirely new research directions. And sort of the combination of all those factors, we were seeing a growing gap between perceived progress and real world impact. And so if you would go to the next slide, please. We're taking a high level strategy that's really like anchored in open science and open source to try address this problem. And so we're taking our solution or our strategy. It's centered around these three pillars. So the first is that we want to provide guidelines. Think of these as like best practices for how to evaluate models in drug discovery. This is not something we can do by ourselves. We don't think we have all the answers. We don't think anyone does. So the way that this is taking shape is that we're forming these steering committees with representatives from various different industrial partners, and then we're bringing them around the table to write up a proposal of like best practices for how to evaluate models. Now, importantly, we realized quite early on that we can't stop there. We need to go beyond publishing papers. We need to make it very easy to adopt those best practices in your day-to-day research. And so we have two other pillars, and one is on tooling. So we want to provide open source software to make it really easy to adopt these best practices in your research. And the Polaris platform, which was used to host this competition, is the primary sort of driving force behind that effort. And then the other thing is community, and this is a very broad category. There's a lot of things that fall within this, or for example, in advanced discussions with academic journals to adopt some of these best practices in their peer review process. But ultimately, it's also the little things, right? Like the Discord community, I think, was a great example throughout this competition of just like trying to change how we talk about evaluating methods, like always questioning our results, trying to really understand why things look better than they are, because they probably tend to be better than they actually are in public or in reality. So like this is a very broad category, but again, like open science, open source, we feel like if we can do these three things, we can make a real dent in this problem. If you would go to the next slide, please. Right. So we've used this platform within the context of this competition. We actually offer other applications, so we can also host benchmarks and data sets. Quite more recently, we also added support for hosting models. Well, model metadata. We're still working on the actual model part. But anyways, it's just to say there's more applications on the platform. I'm sure we've done some things in a poor way. We're looking for feedback, but I do think there's some really cool things there, and we're getting some good feedback. So like, if you're curious, explore the platform a little further. And we also definitely want to run more competitions in the near future. So if that's of interest, please engage through the Discord, and we'll make sure you get a notification once we're ready to host the next one. All right. And then for the last slide, yeah, just if you want to get in touch, various ways through which you can do so. So we actually have an email address at the bottom if you just want to send the team an email, but you can also connect over Discord or just go to the platform or to the GitHub and interact in whatever platform suits your needs. Yeah. So now, truly, without further ado, thanks for the opportunity to just say a few words, and I look forward to the presentations.
Awesome. Thank you so much, Gus. So I think we'll pass to Daniel first. Are you able to
Professor, I'm
switch?
sharing the screen. Can you hear me?
Yes.
Yeah. Hello, everyone. My name is Mohammad Danial, and I am a master student at Shenzhen Institute of Advanced Technology. Today, I will briefly discuss our three submissions to the Polaris Potency Prediction Challenge. This is our team, and in this challenge, we have a total of three submissions. We will discuss them briefly. In the first one submission, our goal was to build target-specific ensemble predictors by integrating multiple chemical representation with diverse machine learning models. So we experimented with three types of molecular representations. The first one is fingerprint-based, like Morgan fingerprints, MICCS keys, and 17 physicochemical features. The second molecular representation approach is graph based, using the GNNs to extract the features from molecular topology and bond structures. And the third one is the large language model approach, where we fine-tune Electrica 125 using the low-ranked adaptation on SMILES. The idea was to capture the different view of molecular structures, one from the substructure patterns, another from the connectivity, and the third from the sequence semantics. In this submission, we used multiple estimators like random forest, light, VBM, MLP, GNN, and LLM regressors. For each representation, three models were pre-trained independently, and the final ensemble took advantage of combination of all the outputs. To reduce the overfitting, we used 5-fold CV and applied early stopping and dropout in LLM fine-tuning. And we also tested the additional modalities like ESM protein features, which we eventually excluded due to minimal gains. And in our second submission, we shifted our focus from only 2D physicochemical descriptors to physics-informed data. So in this regime, we did the molecular dynamic simulation of both protein complexes of 10 and test set in triplets. So first, we performed the molecular modeling on all data sets, followed by the molecular dynamic simulation to extract the key descriptors. We then applied the ASGBIE alanine scanning generalized bond interaction entropy. Actually, this is developed by our lab to calculate the binding free energy or the hotspot residues. Additionally, we conducted the scoring of each MD frames using the Autodoc VINA and performed the trajectory analysis using the CPP trace for contact analysis of each complex. These simulation-based features were combined with standard RDKit descriptors, resulting in a blend of physical and chemoinformatics derived features. While in pre-processing, we applied the standard scalar to the descriptors, and then after that, we used the XG boost and random forest. Sorry. Combined them in a weightage ensemble 60% and in 40% of RF. And after that, sorry, this one, and the mean, sorry. The mean average error for the ensemble model in case of MERS is 0.5183, and still we are working on the search dataset to predict for the search dataset. And these are the some important features which we select for the training and testing. In these, the scoring have negative correlation, while the binding free energy and contact analysis shows the positive correlation. This will be presented by my teammate.
Hello.
You want to share your screen?
Wait a minute. Can you hear me? Can you hear me?
Yeah, yeah.
Okay. Hello everyone. My name is Bohui Xu. I'm a first grade PhD student at Peking University. Next, I will introduce the third part, the multitask multi-model model, based on customized gate control and graph attention for potency prediction. First, in this method, we only use the data provided by Polaris, and aside from the sequences of 2 proteins, no other external data was used. Our method does not rely on any external pre-trained models or external datasets, making it easier to reproduce and deploy, allowing for quick adaptation to other molecular property prediction tasks with better training efficiency. For missing data in the dataset, we did not treat it as a default value like average or median. Analysis of the raw experiment data showed that most of these missing values were either high or low outliers. We filtered out these data points using a customized multitask mask loss function and a mask standard scalar function to complete normalization and loss calculation during training. For prediction in this challenge, we adopted a multitask model based on CGC, which is a multitask model architecture called customized gate control. The model structure is shown in the figure on the right. First, molecules are converted into graph representations. A molecule's graph representation includes not only the node information from atoms, but also edge information from bonds. We also included molecular fingerprints and physical chemical properties like TPSA or rotatable bonds into the molecular representation. Next, the molecular graph passes through an expert layer, which consists of shared experts and task-specific experts. Each expert is a 2-layer graph attention network. Under different tasks, the experts can learn different molecular representations, which are weighted and output by the corresponding gating layer, and then undergo graph pooling to obtain task-specific molecular representations. Now that we have molecular embedding vector, we need a protein embedding vector to better predict molecule-protein interactions. To obtain protein embeddings, we use the ESM series of models to convert the protein sequences into embedding matrices. The molecular representations from the previous step and the protein embeddings are fused through a cross-attention layer. After passing the fused vector through the task head, the predicted value for that task is obtained. Since this challenge uses MAE as the evaluation metric for model performance, we chose L1 loss during the training to enable direct gradient optimization. During training, we employed a weighted L1 loss. We performed TSA dimensionality reduction on the Morgan fingerprints of the molecules in the training and test sets, and results are shown in the figure on the right. It can be observed that both the training and test set molecules are dispersed into many small clusters, but distribution within each cluster is uneven, with many clusters containing only training set molecules and many other clusters containing many test set molecules. To optimize training performance, we calculate the average Tanimoto similarity between the Morgan fingerprints of each molecule in the training set and those in the test set, normalize it, and use this similarity as the weight for the L1 loss calculation. This approach helps balance the data between the training and test sets, thus improving model performance. Additionally, we used Optuna to optimize the model's hyperparameters, such as the hidden dimensions of each task-specific expert, or the method of graph pooling. A total of 4,000 trials were conducted. We performed predictions using the above method and submitted the results. Ultimately, we achieved first place in the Potency challenge with prediction accuracy ranking second for MERS and sixth for SARS. Moreover, this method ranked in the top 3 in both the ADMET and Potency challenges without using any additional data. We conducted ablation studies to evaluate the impact of 13 models on model performance. In the figure on the right, our method is labeled as A. As shown, adding protein embeddings and weighted loss significantly improves modeling performance. The CGC architecture combined with molecular fingerprints also far outperformed single-task GNNs. Furthermore, this model's performance is superior to methods that predict small molecule protein binding pockets using boats and then apply GNNs to the pockets. It also outperformed some pre-trained models like Unimo-2. In the future, to further optimize this model, we need to expand the dataset. For example, we could obtain protein ligand binding energy data from ADB-bind for pre-training and then fine-tune the model using small datasets like dataset of this challenge. Alternatively, we could distill training data directly from high-performing large pre-trained models or docking softwares. To improve the model's generalization ability as the dataset grows, we also need to further increase the model's complexity. For instance, we could adopt the new PLE, which is Progressive Layer Extraction architecture. This architecture essentially deepens the expert layer of CGC, enabling the model to more accurately capture features required for different tasks, while reducing negative interference between tasks. In conclusion, the ensemble predictor, physics-informed machine learning model, and multitask multimodal framework represent our team's diverse approaches to potency prediction. Together, they reflect our commitment to advancing robust and adaptable solutions for molecular property modeling. Thank you for your attention.
Thank you all. Do we have any questions from the audience? Feel free to just unmute yourself and ask straight away.
Hello.
Yes.
Hello.
Yeah, I have a question. So you used 3D structure. So did you consider the protein ligand interactions into this machine learning model, or just you use a 1D or 2D information of protein and ligand?
We just use the smiles.
So
Didn't
that
use
means
any
you
3D
used,
structures.
so that means you used only 1D informations.
We use the smiles to generate the graphs and the let me see, and the fingerprints and the physicochemical properties. So these models and are put into this CGC model.
Yeah, that's true. But I can see in one of your slides you had a miscode training set and test set. And nowadays, yeah, means there are a lot of GNN method is already available with the 3D informations where you can generate the voxel representations of a binding site with particularly certain M strong around the ligand, and then it can be used for machine learning techniques, and that give a more accurate predictions for binding affinity.
So we only use the graph attention layer. We haven't tried other graph neural network yet.
Okay, no, like I just was curious. Means did you use or not? Okay, thank you. Yeah.
I have a question for you guys. So a lot of this architecture is quite deep, right? Like there's a lot of modules that pass to other modules. Did you find you had to do a lot of like hyperparameter optimization to get it to work? Like, did you get a lot out of your hyperparameter optimization, or was it fairly easy to train just out of the box?
Hyperparameter optimization. We just took about 4,000 trials. It's cost about 12 hours or about 12 hours.
And did you see a big improvement over the optimization?
There are some improvement, but not as high as like weighted loss.
Yeah, okay, that makes sense. Thank you.
Can I ask a question, please?
Yeah, of course.
The first speaker mentioned molecular dynamics. I wasn't quite sure what properties were incorporated and how they were incorporated into the model.
Yes, after molecular dynamics, we did the binding free energy analysis, and also the total contact analysis, and along with this, the rescoring of that molecular dynamics trajectories. So we use these three in our training set and also in our test set, these three features.
So, oh, they were three. How did you choose those three features? Because they were the most significant. Yeah. Mm-hmm.
We select the
Okay.
SGBI binding free energy. So it actually based on the MMGBSA. So first we select that one, and also the contact analysis, like how much the ligand has the native contacts with the uh, nearby residues. And the third, we did the autodoc vena has the good results, so we select the autodoc vena.
And later on, you talk about, I think you said your model did very well in the ADMET and the potency, but maybe then that means it did less well on the dock imposes. So if the dock imposes were better, your whole model would also have been better.
The third one, we didn't use the molecular dynamics data. So
mm-hmm.
maybe Lufei will answer it.
Okay, thanks.
All right. I think that brings us to our 5 minutes of questions. Feel free to keep asking any questions you want in the chat. But I think we might pass to Alex from Inductive.
Great. All right, let me share my screen. All right, if you're not seeing a full screen presentation, somebody please give a shout. So I'm Alex Rich. I'm a machine learning engineer at Inductive Bio. I'm excited to be chatting with you all today. First wanted to give a big shout out of thanks to the Polaris team, to ASAP and OpenADMET for organizing this competition. So today I'm going to be sharing a little bit about how Inductive Bio approaches ADMET modeling, both for this competition and then just more generally. And I'm also going to use a big chunk of the talk to walk through some additional analyses that we've been doing after the end of the competition using the unblinded data, as well as actually some additional data from our ADMET consortium and sort of what that tells us about the competition and these types of competitions generally. So first, kind of jumping into our approach. So for context, Inductive Bio provides a suite of ADMET machine learning models that are used by chemists across biotech. And to do this, we are training our models using a pre-competitive ADMET data consortium that is made up of IP-protected data from a number of different companies, and then also public data, and finally, data that we are generating internally as well. And this data is then powering this suite of machine learning models, including the ones that we used for the competition. So we're continually kind of updating and improving our modeling approach. Currently, for representation learning, we're using an architecture that uses message passing graph neural networks that use custom atom and bond level input features. But on top of this, we've added a lot of elements to this architecture and to the training protocol to help us deal with the kinds of complexities that come with just dealing with real-world assay data generally, and then working with a consortium of data that's coming from a lot of different labs and sources specifically. So that includes some modules that basically help us adjust for heterogeneity across real-world data sets. Also some modules that help us deal with assay boundary conditions. So for example, when you get back a metabolism measurement that maybe just says, less than 5 mils per mg per kg, but you can't say more specifically than that what the clearance is. It's kind of just outside of the range of the assay. So it helps us deal with that. And then finally, some approaches for improving our sample efficiency when working with kind of new chemotypes or activity cliffs, et cetera. So we train our models on kind of groups of conceptually related assays. So for example, in this competition, the HLM and MLM predictions are coming from the same base model that has been trained on HLM and MLM data, but also rat liver microsome data and hepatocyte data from a number of species. And then the permeability model in turn is being trained on MDCK data, both MDR1 and wild type, but then also CACO2, PAMPA, and being trained on efflux ratios as well as A to B permeabilities, and so on and so forth. And so each of these models is being trained with kind of separate heads for each of these prediction tasks. And then I think a final thing worth noting here is that we're using a kind of multi-stage training process where the models are first kind of trained on all available data, and then are further fine-tuned to a program of interest, for example, the ASAP dataset. I think something that kind of bears noting here is that we've seen that a real key to finding architectures and training protocols that work well is having really rigorous evaluation. So this is another place where being able to work with dozens of data sets across from dozens of different active programs across a broad chemical space is really valuable. So when we're considering model modifications, we can run lots of model trainings over temporal splits of different programs and series, and that allows us to ultimately make plots like this one at the right. So this is just an example showing kind of a baseline model and 2 experiments where we can see for all these different series that have various different hepatocyte or microsome metabolic stability assays, kind of how each of these 3 modeling approaches fared on different metrics that we care about. And then we can kind of roll that up in a count-weighted way to get overall improvements or degradations in different assays and just sort of overall for the metabolism model. And so what this type of evaluation approach lets us do is just get a really clear sense when we make a change, is it helping in 1 or 2 programs due to maybe random noise or some quirks of that program, or is this really something that generally works across a broad chemical space and will help our models be better? And so kind of in the context of the ASAP competition, while we did sort of create a internal validation set of ASAP data just to kind of confirm that the models were behaving reasonably, for the most part, our hyperparameters and model architecture were selected via this kind of approach across many programs, rather than basing them just on the ASAP validation set, which, while it's kind of the right chemical space, is ultimately going to be a fairly small data set and therefore have a fair amount of random noise if you're trying to optimize just based on that. So that gives a bit of a taste of how we approached the ADMET modeling here. Wanted to now take sort of a closer look at not just how our submission performed, but also what we can kind of learn generally from the other submissions and their reports, and then also what we can learn by comparing the ASAP data to some other lead optimization programs. So I have kind of 2 main lessons that we're seeing here that I'll kind of walk through in turn. But just to set the stage a little bit, in order to sort of be able to compare across different ASAP prediction tracks and then also compare to other programs in the consortium, we built a couple of additional simple baseline models that we shared publicly on GitHub. So those can be found at this link at the bottom. So these 2 models are a descriptor baseline, which is just basically all of the RDKit descriptors followed by a random forest predictor, and then a fingerprint baseline, which is the Morgan count fingerprints with a support vector regression using a radial basis function. These are intended not to be sort of the best traditional ML approaches possible, but just sort of sensible baselines that you might see in a paper or just being used in practice. And so again, you can sort of find the code here and its application to the ASAP test, the ASAP data set, and you'll see below how we kind of use this in the following analyses. So I think the first learning for us looking at the results of the competition is that using additional task-specific ADMET data really was quite helpful to getting top performance. So when we look through the reports, we see that 8 out of the top 10 entrants made use of additional ADMET data. So if we look just for entrants that only use the local training data, we can see that the best sort of pre-trained deep learning model that took this approach was the fifth place result, which is sort of 23% higher error than the best approach using external ADMET data. And then the best kind of traditional ML approach, this was a tree-based model, is 41% higher error. And so we're seeing some evidence from this one program that even though additional ADMET data is usually coming from pretty different chemical space than what we're seeing within the ASAP data set, it really is sort of helping do well on this task. And then finally, we have at the top our two fingerprint and descriptor baselines that are lagging a bit behind the best local ML models. So again, showing there is some benefit to kind of really taking some care and doing that hyperparameter tuning and feature optimization, even with traditional ML. There's definitely advantage there. So another interesting trend that we observed here is that it looks like there's kind of less benefit from large-scale pre-training on non-ADMET data, so using sort of non-task specific data. That best pre-trained model that I mentioned before is using sort of an entirely unsupervised pre-training. But there were a couple of submissions that did pretty large supervised pre-training using either public data sets like from Chembl or PubChem, or also in some cases, large data sets of QM data. And so that's represented by Mole and then also MALGPS. And then one third submission I thought was interesting, obviously a little bit different, but I'm going to bucket it in here as well, is one fine-tuned large language model, which obviously is not really chemistry specific, but is about as massive pre-training as you can get. And so what we're seeing across these sort of non-ADME specific large supervised or large pre-trainings is that they're very competitive with the best local ML model, but aren't clearly far and away beating it, and are not really competing with the best models that really focused on using external ADMET data in the training. And if we jump over into the potency side of the competition, where we can also run these baselines, here, Mole and MALGPS came in first and second place. But what it kind of appears to us is not so much that these models are better in potency than an ADMET intrinsically, but that in potency, there really wasn't this availability of task-specific, so in this case, like target-specific additional training data. So you really just had to rely on the provided ASAP data. And so here, MALGPS and Mole performed a bit better than the best local ML submission. But we're actually seeing that our fingerprint baseline is pretty much neck and neck. It is a little bit worse, but very close to these first and second place submissions. So now there's a really big asterisk here that this baseline was not actually submitted in a totally blinded way to the competition. But still, I think there's kind of a takeaway here that these types of massive pre-trainings are at this point not showing the kind of clear benefits in chemistry that we're seeing in some other ML fields. I think there's good reason to expect that that will be like rapidly changing over the next couple of years, but it seems like there's still kind of work to be done to bring us into kind of that phase of machine learning that we're seeing in other places, and that right now having that ADMET or task-specific data is really, really valuable. Okay, so we have here a couple of overall kind of takeaways that we're seeing from this competition. Something that we were kind of trying to think about is, how much do the conclusions drawn here, would they apply if this competition was run in a different program? And so to get a sense of that, we actually took some different programs from the Inductive ADMET consortium and ran a kind of simulated competition with our two baseline models. So we identified some programs that had either HLM or MDR1 MDCK or kinetic solubility data, and then split the programs into training and test splits using a 75-25 split, and then just trained these two models. So I can give you a sense of what we saw when we did that. So I'll kind of walk through this graph because I know it's got a lot of pieces. So on the x-axis here, so this is for HLM. Here's our different programs. And for each one of them, we have the MAE in log space that was achieved. So for the descriptors baseline in blue, and for the fingerprints baseline in orange, with a standard error determined via bootstrapping. And we've got the Polaris, the results on the ASAP data set for kind of comparison here as dashed lines. And I've kind of ordered these based on the MAE just for kind of visual clarity. And we've got the Spearman Rs on the right. And so what we can kind of see here is that there's a lot of widespread in both kind of relative and absolute performance as you go into different programs. So for HLM, the fingerprint model performed better for most programs, but there are some programs where it's 5% better, and then some other programs where it's 25% better, and then even a couple where the ordering of the models is reversed. And actually, we have sort of statistically significant results in both directions about which model is better. So those are kind of shown with these asterisks. That's at the 5% level and is not doing kind of a multiple comparisons adjustment, because we're sort of imagining what if we just ran this within a single program and kind of drawing a conclusion based on that. So the takeaway here is just that chemical space is really diverse, and that matters a lot to how these things pan out. And so we have here, so here when we look at HLM, the Polaris data I think looks qualitatively relatively within the kind of norms and pretty typical, I would say, of what we're seeing across different programs in terms of performance. But interestingly, when we look at MDCK, MDR1, and at solubility, we actually see the ASAP, the models trained on ASAP, their metrics looking a little bit more extreme. So for MDR1, MDCK, the Spearman Rs that we're seeing from our baseline models on ASAP are actually quite a bit higher than what we're seeing for any of the other programs. And then for kinetic solubility, the MAE is among the lowest that we're seeing for different programs. And so an interesting question that kind of raises is, why is that? Does that sort of speak to unique attributes of the ASAP data set? And when we dive into the test set, we can see that there are some pretty clear reasons that those are patterns that we're seeing here. So when we look at the molecules in the test set, we can see that, first of all, there's sort of two main series or subseries that we see here. So there's one that has this kind of distinctive spirocycle that these molecules tend to be a little bit larger. We're calling that series 2. And then there's series 1 that lacks that spirocycle. And on permeability, those two series behave quite differently. So the series 1 compounds tend to have permeabilities around, say, 25 to 30 microcentimeters per second, whereas for the spirocyclic series, it's a much broader range. And so once the model basically sees that in the training set, it has a relatively easier time ranking compounds at test time, because if it sees something in series 1, it has a pretty good chance that it's going to be higher permeability than series 2. In the kinetic solubility, on the other hand, there's a bit of a different explanation going on. It simply is that almost all of the compounds from both series have quite high solubilities, right up against that 400 micromolar experimental limit of the assay. And so the models can typically do a very good job of just saying, hey, this is going to have a solubility of around 400 micromolar. And so MAE is quite low, whereas ranking within this data set is pretty hard. And so really what I think this is exemplary of is that having competitions run on real program data is in fact quite important, because these types of quirks and nuances basically universally happen across any real drug program. And we want to be evaluating and capturing how models do in these real world settings, rather than on sort of a very artificially constructed data set. But of course, the thing is that every program is going to have its own quirks and nuances that are unique to that chemical space. And so then to kind of wrap up here, so what we're seeing again is that the results of this antiviral competition are suggesting that additional ADMET data in training is really beneficial, whereas this more non-task specific massive pre-training is less beneficial. But again, comparison across programs really reveals how much this can vary based on the chemical space. So really what I kind of want to conclude with is, again, the idea that like it would be really great to keep running more of these. So, Polaris and ASAP and OpenADMET have done a really huge service to the community by running this first competition. I'm sure it was quite a lot of work. But yeah, as Cass was saying, it would be really great to kind of keep running these so that the community can get kind of more windows into what works in different parts of chemical space. So yeah, thanks for listening, and yeah, happy to take questions.
Awesome. Watch this space maybe for more competitions, but no promises. I need to speak. Questions for Alex?
I have a clarification question. What does non-task specific mean?
Yeah, so here it means like not directly relevant to the assay being evaluated. So for example, with the MOLE and MOL GPS, they included, QM calculated data or lots of, I think, mostly potency-based bioassays from Kemble, but didn't include ADMET data in that pre-training. So that's what I'm saying by non-task specific pre-training.
Sorry, can you just elaborate? What's the premise of including that? Is there some sort of trying to precondition the model to know what molecules are, or what's the point?
Of doing the sort of general pre-training?
Yes.
Yeah, I mean, I don't know if there's any of the authors of sort of those approaches here. I'd love to hear their answers as well. I do think ultimately these types of approaches seem quite promising to me, that if you train models on enough data from a lot of really broad properties, that hopefully the models eventually just kind of like learn chemistry, and then they can do well even on a unrelated task. But at present, our models are kind of taking this approach of really focusing on the task-specific training, because at this point we're seeing that really that is what's bearing fruit, and that we're not quite there yet in terms of this idea of bringing in other types of data having really big dividends. So I would say, again, hopefully that's something that will probably evolve in the future, but so far has not paid as big dividends as we might hope. Does that answer your question?
Yes, thank you.
Alex, just a follow-up on that question. Why do you think that is the case that in chemistry, the pre-training isn't kind of paying the dividends that is paying in other fields like imaging and so on?
Yeah, that's definitely $1 million question. I don't think I have a really great answer for that. I will say, like, I mean, I think chemistry is, everyone here will say it's just a really hard, complex space where chemical space is really craggy, where very small changes to a molecule can have massive changes on the molecule's behavior. It's a bit harder to do, I think, good unsupervised training in chemistry than in something like language or vision, for example. So I think that, yeah, there might be a number of reasons why it's been a bit trickier to have kind of the big breakthroughs that we've had in some other machine learning fields. But yeah, I definitely don't have the answer.
How do you curate the data for the assay endpoints that you use when across pharma labs have tried to compare data? Sometimes it's difficult to kind of aggregate the individual results into kind of a coherent whole.
Yeah, I agree. It's definitely a really difficult problem. We use sort of a lot of different methods here, some on the more sort of manual curation side of making sure we're kind of collecting assay protocols from all of our partners and understanding really deeply how those assays are different, and making the correct adjustments, or keeping things sort of in separate tasks when they're sort of not really directly comparable, and doing the same when we're sort of going through public data sets that we're pulling in. So there's a big part for that kind of manual curation. And then there is sort of a role for the architecture itself, as I mentioned, where we've kind of built some modules in to our architecture that can kind of keep track of the fact that data is coming from different labs, different assays that maybe should be somewhat commensurable at a high level, but we know that there's going to be sort of nuances to how things are done in different places, and that data might be slightly different, and the model can kind of learn to account for those differences in its learning process.
Cool, thanks.
Mm-hmm.
I know the author of Mole GPS is somewhere in the or was somewhere in the audience. I might give them right of reply if they want to speak up, but no pressure.
Hey, thank you. Yeah, I'm not really the author of Mole GPS. The authors of Mole GPS are in Balance Labs, and I don't know if some of them is in the audience. We just use it. That's a clarification on the case of Mole GPS. We didn't fine tune the whole model. As Alex says, the idea is to have a foundational model that it can be then applied to a specific task and retrain. But here we want to try to see only use the embeddings, so the representations, to train a very, very small neural network, a full connected neural network that have like 5 layers. And we did just a very short optimization, just like 100 trials of tuna. And that was basically the submission. It was a very, very short project. We put it together in a few hours. Really.
Yeah, that's really interesting, like additional context to have. And I do feel like in general, I feel like very positive about this sort of general approach, being something that's going to sort of drive these breakthroughs for the community over the next few years, but just trying to take this kind of critical take of like, how much benefit are we getting from it right now, and where do we kind of need to kind of keep pushing that forward and finding the way that using these big models works best.
That makes sense. Cool analysis, by the way.
Mm-hmm.
This is all very fantastic discussion, but I might have to cap it there to pass to our last speaker. Thank you, Alex.
Great.
All right, lucky last. Will?
All right. Hi everyone. Let me share my screen. Let me know if you guys can see it. All right. Yeah. Yeah. Hi everyone. My name is Will. I'm a machine learning scientist or engineer. So I'll be talking about my learnings and the approach that I use to solve for leak and post challenge. So you can see from the title that the high-level methodology here is to leverage the Boltz model, which is like the reproductions of AlphaFold 3, but very but some modifications that we believe that it's kind of better. So all right, so high-level overview of leak and post challenges. So we want to basically predict the leak and post that binds to 2 proteins here, SARS-CoV-MPro and MERS-CoV-MPro. But for the challenge in the training data set, there's only PDB structures of SARS-CoV. MERS-CoV is basically blind. So I was thinking that, okay, if like there's only like one rigid protein structure and some like ligands, and we want to know how to bind protein, we want to model a protein ligand interactions, maybe docking is like traditional docking or like more advanced docking would be enough. But in this challenge, like is a little bit more than that, where the protein structures of MPro is also not given, even though like we can also find some similar structures for the protein data bank. But I decided to use like a little bit more complex model to model not only just a docking protein ligand, but also like generalized biomolecular interactions. So I chose Boltz 1, which is like the model developed by Gerber and Jeremy from MIT lab that basically proven to have a very comparable performance with AlphaFold 3. So, but the methodology here that I further developed from just using the Boltz-1 purely contains some data set preparations on fine-tuning the Boltz-1. And the key takeaways here is, okay, first, the results show that like the performance is like not the best, but it's like around like 84% success rate in the benchmark, where the best score is 86.4%, which I believe that the performance gaps here could derive from some limitations on my fine-tuning pipeline, like that could lead to underfitting to blinded protein, more overfitting to the unblinded protein. And yeah, fine-tuning also pipeline that I developed here is not totally the best because of some computational efficiency limitations, because it takes time to train a model and learning the structures over time. So another key lesson is while working on a ligand pose challenge, I learned that like PDB structures can have a lot of nuances, and working with that, evaluating the results can be a little bit more challenging than expected. So I'm gonna talk more about that in the later slides, but yeah, so basically sometimes the predictive structures are not aligned with some domain knowledge. Like for example, it doesn't respect the physical constraints where you have like triple bond. Sometimes like the predicted structure is not totally linear on that SP hybridization. So I think the Boltz-1 or AlphaFold 3 architectures can be like very complex. So I'm just gonna skip the most complex part and only focusing on the part that I further develop from the existing architecture. So yeah, I think basically to train like AlphaFold 3 or Boltz, you need a lot of computational resources and full training on all structures and confidence modules of the architectures. It can be very memory efficient, and I also like kind of developed like a parameter efficient fine-tuning to kind of to make like a trade-off of the performance with the speed that I can finish in time and make the submissions to the challenge. So basically the parameter efficient fine-tuning here just contains basically a roller and some adapters in the architecture. So generally like the Boltz-1 architectures has a lot of layers inside and contains a lot of LoRa linear embeddings. Which are common layers that we can fine-tune with row-length adaptation. But I kind of focus also like one of the layers that are commonly used in this entire structure is attention pair bias, which is basically the representations of atoms or protein tokens. And you have like these kind of atom or token representations, and you have also the pair representations of it, which are you use like basically a relative distance between atom coordinates or a token or residuals of the protein, and you basically use this as the condition embeddings in the diffusion models. So in both one, like basically you can think of like there's a noise of coordinates, and we're trying to learn some representations of those atoms and tokens and proteins in the space, and we use that to diffuse all of these noises and denoise that to become like the richest structures, the final structures. So in this, that's why attention pair bias is a very important layer in doing like to understand basically the structural inductive biases of the molecule. So and I added like a lot of fine tuning on that. Basically, you can see QKV here represents like a basic basic attention mechanisms. So I kind of used reduce some computational expensiveness there by adding roller there as well. And along the way, you can see this is like adding some biases, and there are some gate layers that we use to adjust the attention scores or the weights before we returning the final representations of the pair. So the green layers, which are like basically a fine tuning components that I adjusted or replaced in both bond architecture. So and yeah, basically I use this to kind of basically fine tune the model in order to get like the results more efficiently with like one like a single GPU training maybe like 1 or 2 days on 200 structures around 200 structures in the training data set. And yeah, so as a baseline, like both one is trained on like maybe around, I'm not sure about the exact number. It's written in a paper by the team, but it's trained like using 880 gigabytes like for about 2 weeks or something, 20 days, which are very expensive. Yeah, so further than that, like just like like adding fine tuning layers in the architectures, I also like implemented some denoising mechanisms to improve like the way that we are trying to interpolate those coordinates from the noises to the structures. So there's a technique called auto guidance, which are basically using the worst version of the model to kind of guide the way that these coordinates are distributed on the space. So I found that like adding this, particularly in this challenge, wouldn't be very helpful because we are focusing only on the ligand posts. But this also can be very useful if we want to model or predict like larger molecules, DNA, RNA, or like a larger protein, longer protein residues. Yeah, this technique was actually also used in the recent model from NVIDIA called Proteina to scale up the protein backbone generation. And this can be very effective because it kind of balances the benefits of diffusions between conditioned guidance and unconditioned guidance. So what I mean by conditioned guidance would be like some prior knowledge that we are injecting into the model predictions. For example, if you know that this is like a protein, alpha helix protein, beta sheet proteins, we use that to kind of approve the condition guidance here. So in our case, like Bodes one uses mostly atom level and token level pair representation as a condition embedding step. So I think that's why it's kind of mostly focused on condition guidance diffusion already. So yeah, so I think this is a little bit more too mathematical, but I'll switch back to kind of the way that I use specifically for this challenge. So when you use Boltzmann architecture to make predictions, you also can basically like you are trying to explore how the coordinates can be diffused into the rigid structure. And there's so many ways of that, like in 3D space, there's so many ways of these coordinates are diffused. And you could also set a parameters called the number of samples that you want to predict for that for a particular data point for a particular pair of protein ligand. And the more samples that you predict, the more chance you have, the more likely you are going to predict like a more accurate pose or structure. So yeah, sometimes like it's not the best. What in one shot is not going to give you the best model, the best structure. But if you do it more, you have more probability of doing that. And that, but basically, out of these like number of these many predicted samples, how do we rank the final structures that we want, that we believe that is the best? So there are a couple of metrics that we consider at Boltzmann inference here, including confidence score, TM score, protein ligand IPTM score. What do they mean exactly? Confidence score is basically how much the model thinks that this is actually reliable. These predictions are reliable. But also TM score, IPTM, are basically use like predicting like the alignment of predicted structures against the template. And protein and ligand specifically focuses on the interface level, the predicted coordinates at the interface level. So even though like we have a final confidence score, it doesn't necessarily mean that the structure with highest confidence score indicating the lowest RMSD, like the most accurate structures. So I kind of do some correlation study of like which metrics could also lead to the lowest RMSD here, even though these scores can be highly correlated. So for this challenge specifically, since it's like a ligand pose, so I chose the samples with the highest ligand IPTM score as a final submissions. It would be very interesting to see how to kind of do further analysis on like, okay, how the structures with highest confidence score or highest like a protein IPTM score how much would it differ from the one that I sent it in the submission. So yeah, I think that's pretty much it in the methodologies, but let me kind of do some advertisements for both community here. So actually Boltzmann, it's not only just the reproductions of AlphaFold3, but also some further improvements in interpolations. And there's a Slack community of scientists who contributed to this repo, led by Grebel and Jeremy from MIT lab. And yeah, so recently both has been developed to Boltzmann-X, which uses like even stronger, reliable protein ligand denoising, the specific denoising mechanisms for protein ligand interactions, which uses like FK steering to improve the predicted structures with higher physical validity. So as mentioned earlier, that sometimes like the predicted structure doesn't respect like a hybridizations of the bond, but or sometimes the protein structures and ligand can overlap because of the training data. But with this denoising mechanisms, like there's a big improvement in the physical validity here. And also like Bose1X incorporates more memory efficient form kernel optimization developed by the scientists in the community, which are probably more efficient than my fine-tuning pipeline now. And also it's like, like FK steering denoising is used mainly for ligand to kind of predict the not only for ligand but also like for physical constraints here. But I would say sometimes if we look at the physical constraints and structures, it could outperform fine-tuning techniques as well. But it would be very interesting to see how non-fine-tuned model with this denoising mechanisms would be compared to the fine-tuned version. Yeah, and like if this new denoising outperforms the fine-tuning, it indicates like a very promising improvement. Not only just like domain experts or can just use anything at inference and not just retrain or rebuild a wheel in the training step. So yeah, I'll be open to questions now, but yeah, I have my fine-tuning pipeline for boats in my GitHub repo, and I also have like an MCP server, which is like a very new type of thing, and tutorial as like agentic interface where you can chat with the fine-tuning pipeline. You can say that, okay, I want to fine-tune this and that, and I want to connect this part to that part to process this data or analyze the results. You can do so by just chatting with this agent. Yeah, I developed that. It's still in a lot of things are still in work, still work in progress, but it would be very good if I get any feedback for the community. And yeah, lastly, last but not least, I will advertise for the both community here, and if anyone is interested, feel free to join from this link. I will. I think I can share this slide with the group as well. Yeah, I think that's it. Thank you.
Awesome, Will. Thank you so much. Do people have questions for Will?
I'll ask a question. So in drug design, often we have activity cliffs. So two molecules that are extremely similar but have a very varied biological activity. So do you think your model will be sensitive enough to distinguish between why one molecule is less active than the other because of a certain interaction, a lack of space in the cavity, or is it asking too much?
So I think it depends on how hard that bioactivity cliff is actually challenging the model. If the molecules are almost identical, I would say this model may not capture that because remember, like we are this both one mostly represents the struct like atoms or tokens, and use that to kind of diffuse the structures in 3D space. We are not using like MD simulations and things like that, but I would say combining both one with maybe MD trajectories would be better for that.
Thank you.
I have one question.
Sure.
Does your model is useful for like PROTEC? I mean, for tertiary protein structure designing, which is like in use in size like more than 4,000 amino acid sequence.
Like a very long
Yeah, it's a protein tertiary protein structure, so which means usually like three homodimer means attached together, and is it useful to means build this kind of model?
It is possible to model ternary protein structure, but there's a limitations on the sequence, the length of residuals that you put in at inference time, because if it's very long, it can be very computational expensive. You need like very high performing chips to do that operations. It can be in memory inefficient. There can be memory bottlenecks there. But I think generally for ternary on bigger structures, there isn't like I would say there isn't like a comprehensive studies or a benchmarking for boson against other models. I think it's like recently, like I also used boson to make to like try to predict some ternary protein structures, and I can see some limitations there. I believe that there is room for studies and research on how it actually performs in terms of quality against the benchmark. Yeah. Does that answer your questions?
Okay. Yeah, thank you.
There's just one more question in the chat, Will, around predicting 3D structure of oligonucleotides like DNA, RNA, things like that. Are the Boltz models performant in that kind of domain?
Yeah, I think in DNA and RNA structures, or I think this is like also quite similar to AlphaFold 3. There's in like a significant improvement from AlphaFold. But I try to also add some prior secondary features in my repo here, like to deal with the DNA and RNA MSA. Yeah, I think that's still in work in progress to kind of conduct some benchmark studies on how it actually helps that. But basically, both one now doesn't currently support like DNA or RNA specific features, and I think that's a big room improve. There's a big room of improvement there.
Awesome. I think that might bring us pretty much almost exactly to time. I wanted to take a chance to thank all of our speakers so much for taking the time to share your expertise. It's so interesting seeing all of the amazing scientific work that's been done. So thank you to all of our speakers. I'm doing a little virtual clap here on behalf of everyone, but and thank you to all of the participants, especially those here today. We'll be sharing this recording. Hopefully, so you can let me know if parts of your presentation cannot be shared. Just let me know. But in maybe a couple of days to a week, we'll share this recording on YouTube and also on the Polaris Slack. So thank you all for taking the time. Have a wonderful rest of your day. Thanks all.